Hi friends, today we are going to start a new subject, that is biotechnology, or you can say principles of biotechnology. Friends, this is very important. Mm, so friends, without wasting more time, let us start. The term biotechnology was coined by the Carl Ehrlich in 1919. Now, what is biotechnology? So, the biotechnology is the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. This definition was given by the OECD, means the Organization for Economic Cooperation and Development, in the year 1981. Now the human gene producing insulin has been transferred and expressed in the bacterium like E. coli. Now coming to the history of biotechnology, in 1982, Government of India set up an official agency, that is NBTB, means National Biotechnology Board, which works under the DST, Department of Science and Technology. But in 1986, NBTB was replaced by the DBT, means the Department of Biotechnology. And moreover, on the proposal of United Nations organizations, the International Center of Genetic Engineering and Biotechnology was established to help the developing countries. So ICGEB has two centers, one in New Delhi and other in Italy. Now coming to the abbreviations used in biotechnology. Friends, here there are some of the abbreviations which you have already studied in your class 11th and 12th, like RFLP, restriction, fragment length, polymorphism, cDNA means complementary DNA, mtDNA means mitochondrial DNA, PCR, polymerase chain reaction, GMO, genetically modified organism, ELISA, enzyme linked immunosorbent assay, IAA, indole 3 acetic acid, IBA, indole 3 butyric acid, TI plasmid, VNTR, GESC, EDTA you have also read in class 12 in inorganic chemistry, also in the lesson coordination compounds, and MOET. Now coming to the history of plant tissue culture and genetic engineering. So the plant tissue culture broadly refers to the in vitro cultivation of plants, seeds or plant parts or nutrient media under aseptic condition. Here in vitro means outside. So the cell theory which was given by the Silden and so on, but in 1902 Haberland attempted to cultivate plant tissue culture cells in vitro. So he is regarded as the father of plant tissue culture. Now another very important word is totipotency. So totipotency is the ability of plant cell to perform all functions of development. Now the first embryo culture was done by Henning in 1904. In 1964, Maheshwari and Guha were first who produced haploid plants from pollen grains by culturing anthers of Dhatura. Now coming to the history of biotechnology. So the, as you know, fathers of plant tissue culture was Haberland and Liebach, who used of embryo culture technique in intraspecific crosses of linseed linum. This is written next also. Now Van Overbeek, he uses use of coconut milk containing a cell division factor for the first time to culture Dhatura embryos, and Skog and Kornberg. Kornberg is very important because he synthesized in vitro DNA, in vitro synthesis of DNA, and Skog and Miller again very important. He both discovered a plant growth hormone, that is cytokinin. Now H.G. Khurana is again very important. He awarded Nobel Prize for deciphering the genetic code and also deduced the structure of a gene for the East alanine tRNA. And then again you can see here, as Eli Lilly, you have also studied about this in class 12th, in microbes and human welfare, or because Eli Lilly it was an American company who started commercial production of human insulin, that is called humulin, through genetic engineering in bacterial cells. And then Carrie B. Mullis, this is also very important because he discovered the PCR, polymerase chain reaction. And Alex Jeffrey is again most important because he developed the genetic fingerprinting technique for identifying individuals by analyzing polymorphism at DNA sequence levels. Now here some of the technology which is used in plant tissue culture. Here you can see anther pollen, basic things are there. Anther pollen, embryo, cell, protoplast, batch culture, and some of the important I am mentioning here explant. So it is a piece of tissue which is used to initiate tissue culture or removing shoots from callus, separating individual shoots from proliferating mass of shoots. Now callus. So callus, it is a tissue arising from disorganized proliferation of cells either in culture or in nature. So callus is very important. Now micropropagation is again very important. Micropropagation can be defined as the production of miniature planting material in large number by the vegetative multiplication through growth hormones regeneration. Totipotency means the ability or the property of a cell to give rise to whole plant. This capacity is called, or this potential or this potency is called totipotency. Now amplification means creation of many copies of a segment of DNA by PCR, polymerase chain reaction. Now coming to plant cell and tissue culture. So steps in general tissue culture techniques, merits and limitations. So in plant tissue culture, in this technique, a small pieces of viable tissues called explant are isolated from parent plants and grown in a defined nutritional medium and maintained in controlled environment for prolonged period under aseptic conditions. So there are four main stages. They are initiation of culture, multiplication or subculture, development and differentiation, and hardening. So first one is initiation of culture. The most important factor in tissue culture technique is maintenance of aseptic condition. So here you can see culture vessels has to be plugged and autoclaved at 121 degrees Celsius, and the transfer area should also maintained free of microorganism. Now second point is multiplication. So after 2 to 3 weeks the explant shows visible growth by forming either callus or differentiated organs like shoots, roots or complete plantlets depending upon the composition of medium. Now coming to third point was development and differentiation or organogenesis. So the concentration of phytohormones in the medium are changed or altered to induce differentiation in callus. As friends, you already know that the high cytokinins to oxygen ratio induces shoot formation, and reverse means high oxygen to cytokinin ratio, so root formation. Now steps in plant tissue culture technique. So here first step is selection of plant, then isolation of explant, after that sterilization of explant, and then inoculation of explant, then incubation, initiation of callus, subculturing, regeneration and hardening, and last step is transfer of plantlets to greenhouse or open field. Now fourth point is hardening. Here the gradual acclimatization of in vitro grown plant to in vivo condition is called hardening. Simply means a gradual acclimatization of in vitro plant to in vivo condition. In vitro condition to in vivo, this is called hardening. And here this ratio is important. Clay soil is to coarse sand is to leaf molds is the ratio 1:1:1. Now application of plant tissue culture in crop improvement. So it helps in mass multiplication of plants. Some perennial crop plants like ornamentals and fruit crops which cannot be propagated through seeds. So in such crops micropropagation helps in rapid multiplication. And rapid multiplication of rare and elite genotypes such as aromatic and medicinal plants. Fourth point you can write a screening of large number of cells in very small space. Now fifth point is induction of flowering in some trees that do not flower or delay in flowering. And the most important point is virus free plants can also be produced through meristem culture. And in case of certain fruit crops and vegetative propagated plants where seed is not of much economic importance, so triploids can be produced through endosperm culture. And last is development of transgenic plants. So this tissue culture has some advantages as well as disadvantages. So advantage is rapid multiplication within a limited space, not time bound and not season bound, and free from pest and disease. But the disadvantage is it is very much laborious, costly, special risk is required. And now coming to different types of techniques which is used for sterilization is the media used for plant tissue culture contains sugar as a carbon source. so thereby attracting a variety of microorganisms including fungi and bacteria, and these are toxic to plant tissues. Therefore, sterilization is absolutely essential to provide and maintain a completely aseptic environment. So, but what is sterilization? So, sterilization can be defined as it is a procedure which is used for the elimination of microorganism and maintaining aseptic or sterile condition for successful culture of plant tissue or organs. Now, there are some different types of sterilization. First, as I have mentioned here, dry sterilization, wet sterilization, ultrafiltration or filter sterilization, UV sterilization, film sterilization, surface sterilization, and whipping with 70% alcohol. So, first one is dry. As the dry heat, as the name itself is saying, it means exposure to hot dry air at 160 degree to 180 degree for 2 to 4 hours in hot air oven. Now, second is wet heat sterilization. It is a method of sterilization with water vapor under high pressure to kill all the microbes. Third one is ultrafiltration or filter sterilization. So here some chemicals are filter sterilized by passing through a bacterial proof membrane filter under positive pressure. Fourth is ultraviolet sterilization. Here UV light sterilizes the interior portion of inoculation chamber. Fifth is flame sterilization. As the name itself is saying, metallic instruments like forceps, needle, spatula are sterilized by dipping in 95% ethanol, followed by the flaming and cooling. This is called flame sterilization. And sixth one is chemical sterilization, means culture vessel is treated with an appropriate sterilizing agent to inactivate the microbes present on their surface. And seventh and last sterilization is whipping with 70% alcohol. Here sterilized by wiping them thoroughly with 70% alcohol, and the alcohol is allowed to dry. So friends, rest in next video, and friends, you can comment also that on that next video on which topic I should make the video. So up to then, bye bye and take care.